I'm going to be covering haemostasis today. So in terms of the learning objectives, we're going to be looking at platelet aggregation and activation, stages of haemostasis, a couple of models for the coagulation cascade, common tests, and a bit about fibrinolysis. So haemostasis is really the interplay between the endothelium, platelets, and coagulation. And when we're not bleeding out to death, the endothelium prevents unnecessary clotting from occurring through things like nitric oxide, thrombomodulin, even makes little bits of heparin as well, which is pretty nifty. But if we do run into trouble and we, start bleeding for any reason, there's a kind of 3-step process that our body undertakes in order to help us. We go through vasoconstriction, platelet aggregation, and the coagulation cascade. So in terms of vasoconstriction, as seen in this cross-sectional microscope image, this is handy because vasoconstricting, the smooth muscles react, they constrict, it prevents clots being washed away, it prevents further bleeding from occurring, which is helpful for us. And at the same time, we see platelet aggregation occur as well. So when there is a disruption to a blood vessel, when the endothelium is disrupted, external factors which are not usually exposed to the internal environment of a blood vessel are now exposed. And that includes things like von Willebrand factor, collagen, and tissue factor. And these are responsible for platelet aggregation. So this childlike drawing depicts what may happen if, as you see above, there's a defect in the intima, which exposes collagen, also exposes tissue factor. And so our circulating platelets, which are minding their own business, when they run into these things, they, through their receptors, the glycoprotein receptors, will bind von Willebrand factor, they'll bind to these areas, the collagen. The tissue factor in turn will activate these platelets, which will then start to release their own modulators like fibrinogen, more von Willebrand factor, ADP, which helps to bind more platelets, thrombin, serotonin, thromboxane A2, these can help with more vasoconstriction. And so there's this cascade of platelet aggregation, and they bind together through fibrinogen and von Willebrand factor. And fibrinogen is a sort of weaker precursor to fibrin. It's only made into fibrin when some of the end proteins are cleaved away. But at this stage, there's this platelet plug which is forming. It's not solidified yet through fibrin, but it's there and it's starting to deal with the process of the bleeding. And so whilst that's going on, that platelet aggregation, we also helpfully have a coagulation cascade. And the end goal of all of this is really to have a fibrin mesh to hold all these platelets together to basically form a plug. There's a couple of models to illustrate this, and the one we're probably most familiar with and have the most PTSD from is the 2-pathway model. This is more reflective of what you might see in the lab, but not necessarily very intuitive for explaining what happens, biologically, for which there is a more recent cell-based model which we'll briefly go over as well. But before that, just as a more, that's perhaps quite interesting rather than need to know thing, the areas where we get our coagulation factors from include the hepatocytes. You can see the hepatocytes make the vast majority of clotting factors. Endothelial cells also make a couple. And then the plasma has one floating around too. And to put some names to these, we've got fibrinogen, which is number 1, the big daddy down there. And then prothrombin, number 2. Number 3 is tissue factor. And number 4 just happens to be a calcium ion. And the other factors do have their own, often eponymous names, but you don't really need to know those. So the 2-pathway model. We've certainly seen this before. This is a vastly simplified version, but 2-pathway model basically is 2 different paths that feed into a common pathway which ends in producing what we really need if we're bleeding out, which is a fibrin clot. So to start with the intrinsic pathway, this is essentially a series of enzymes cleaving each other into activated forms of the enzyme to then feed into the common pathway. And the way that's easy to remember this is just to count back from 12, skipping out 10. So 12, 11, 9, 8. So you can see factor 12 becomes factor 12A, which makes factor 11 into activated factor 11 or 11A, and so on and so forth. And finally, activated forms of factor 9 and factor 8 form together into a kind of protease, and they feed into the common pathway. Extrinsic pathway, predominantly two key players, which are tissue factor and factor 7A. So the tissue factor, as we know, isn't really in the blood normally, but when there's disruption to the vessel wall, then it's exposed and it activates the extrinsic pathway, forming with factor 7A to feed into that common pathway. And what is the common pathway? Well, it really starts with factor 10, funnily enough also known as Stewart factor, so we could call him Stewart from now on. So Stewart becomes activated Stewart, or factor 10A, through the activity of tissue factor in 7A and 9A and 8A. Factor 10A then turns prothrombin into thrombin. And now thrombin is a very central, important modulator of all of this, going downstream and upstream. And thrombin, in turn, turns fibrinogen, remember that kind of weakly binding molecule on the platelet plug, into fibrin, or a fibrin clot. Now we have a much stronger end product. And importantly, thrombin also exerts positive feedback to 11 and also factor 8, and that propagates and amplifies the entire process again via the intrinsic pathway. And kind of more clinically, apixaban, rivaroxaban, edoxaban, and I've spelled that a bit weird, fundaparinux might be better. These directly inhibit factor 10A, so you won't get thrombin from prothrombin because that process is taken out. Unfractionated heparin and low molecular weight heparin, they indirectly inhibit factor 10A by activating antithrombin. And antithrombin will inhibit 10A and thrombin. Dabigatran is a direct thrombin inhibitor. So the cell-based model, we're really revisiting my childish drawing and building on that a little bit just to perhaps produce something a bit more intuitive to explain coagulation. So there's an initiation phase, which we've seen. There's an injury and essentially this is the extrinsic pathway really. And collagen is exposed, tissue factor is exposed, and actually there's a small amount of thrombin produced at this point. And so our circulating platelets, when they come into contact with the thrombin and the tissue factor, they're activated. And they create their own thrombin, their own other modulators. And then there's, at this stage, a propagation or an amplification of the cascade. And this is more analogous to the intrinsic pathway because we now have factors such as factors 5, 8, 9 and 11 involved. And remember that's because thrombin has a positive feedback which propagates through the intrinsic pathway. And so now that positive feedback amplifies and there's more thrombin produced, makes up about 95% of this whole process. And then you get just this propagation that continues and you hopefully end up with a nice strong clot. So in terms of the labs, just have a quick look at this and try and remember for a second what lab tests we use to explore the extrinsic and the common pathway. So you've had your second and it's prothrombin time. So a prolonged prothrombin time can be due to a few things, but oftentimes we think of a reduced amount of vitamin K, for example, or vitamin K dependent factors being depleted or inhibited. And that's classically warfarin. And part of the reason for this being reflected in the PT is because factor 7A, which is a vitamin K dependent factor, has the shortest half-life of all the vitamin K dependent factors. So you'll see the PT be affected first before anything else. Hepatic impairment, obviously, because factor 7A is produced by the liver, that can prolong your PT. And DIC as well can prolong your PT additionally. And just remember the other vitamin K dependent factors include, well, they're basically 2, 7, 9, and 10. And INR is a standardized way of measuring prothrombin time that uses a kind of control or standard measure and compares the patient's prothrombin time to that measure to come up with a number. And the way I remember this is the acronym WEPT, or I think of warfarin, extrinsic, and then PT. And if I remember warfarin, I can remember the vitamin K bits as well. So that leaves us with the intrinsic and common pathway. So the activated partial thromboplastin time, quite a mouthful, but that basically measures everything except the intrinsic, so it does include the common pathway. And you might see this prolonged in haemophilias because we know factors 9 and 8. Factors 9 and 8 are haemophilias A and B, or B and A, I can't remember which way around it is. Unfractionated heparin as well, they inhibit factor 9, factor 10, and thrombin as well. So that will prolong your APTT. And if you've got someone on an heparin infusion, you will often use the APTT to guide the dosing for that. DIC as well will prolong your APTT. So now just a bit about fibrinolysis. So eventually we want our clots to be broken down, otherwise that would be bad. And so plasminogen is one of the first parts in this process. And we need tissue plasminogen activator to activate plasminogen into plasmin. Once we have plasmin, we can now start to break down those fibrin clots, the fibrin mesh that we had and we relied on. And hopefully by this point, tissue healing has occurred and we won't continue to bleed. And the fibrin degradation products that we get from that can be measured in the form of a D-dimer. So obviously if you've got a big clot, there will be ongoing breakdown of that clot, such as in a PE, and so you would assume that your D-dimer will be raised. Something like alteplase or streptokinase, something like that, that's basically a recombinant form of tissue plasminogen activator and will speed up this process of turning plasminogen to plasmin and fibrin breakdown. Conversely, tranexamic acid will inhibit tissue plasminogen activator and hopefully prevent further breakdown of a clot. And briefly, DIC, pretty awful process where you get dysregulation of clotting, and one of the primary things that can trigger this is massive surge of tissue factor causing huge amount of clotting and the coagulation cascade, and you will start to use up a lot of your coagulation factors and things, but simultaneously there will also be breakdown of these clots, and so you may start to bleed as well. So you have often concurrent thrombosis and hemorrhage as well. Pretty horrendous. So if we're using up a lot of our clotting factors through so much tissue factor being activated, we'll see a prolonged APTT and PT because we're running out of clotting factors and we take longer to clot. We'll also get a raised D-dimer because we've got so much clotting being formed that we'll get fibrin degradation products. And the fibrinogen being used to form those clots is being used up so rapidly that we'll have a reduced or a low fibrinogen. And obviously we'll have a reduced amount of platelets as well. So that's really it. I'd really appreciate if you could give some feedback. Hope that was helpful. Thanks very much.